Yes, okay. Uh, I request the MC to start now.
A very good morning, everyone. I hereby welcome you to the third day of the international webinar, The Modern Trends in Microbiology, organized by the Department of Microbiology of St. Xavier's College, Kolkata, Autonomous. I would now request the head of the department, Dr. Shudeshna Sham Chaudhary, to introduce our speaker for the first technical session, Dr. Bharat Vedyanathan. Ma'am, over to you.
Thank you, Moitri. So, very good morning, everyone. It is my great pleasure to introduce Dr. Bharat Vaidyanathan, who is our ex-student of Department of Microbiology, St. Xavier's College. I can remember that Bharat was very sincere and hardworking student. I'm feeling proud to introduce Dr. Vaidyanathan and talk about his research career. He has more than 10 years of scientific research experience with special focus in immunology and microbial pathogenesis. He completed his graduation from St. Xavier's College Microbiology Department. Then he did his master's degree from CU, Calcutta University, Biochemistry Department. After that, he had pursued his PhD work in immunology and microbial pathogenesis from Cornell University, USA. He was involved in postdoctoral research work in Department of Immunobiology, Yale University, USA. He has major contribution as research analyst for supporting biomarker team for drug repositioning. He has many publications in peer-reviewed journals. He has expertise in vitro human B cell differentiation and functional assays. He is involved in various multi-omics projects. He has received many prestigious awards, including Outstanding Performance Recognition Award at EMD Serono R&D Institute, USA, where he is now currently Senior Scientist in Exploratory Immunology. His today's talk will be focused on virology meets immunology through an evolutionary lens, rheumatic drug, repurposing for COVID-19. So once again, welcome Dr. Vaidyanathan. Thank you.
I hope everyone can see my slides.
Yes, sir. Yes, sir.
Thank you very much, Dr. Sudeshna, for the kind introduction. It is really a privilege for me to deliver this webinar at my alma mater, which is very close to my heart. I am feeling very nostalgic. The reason being that I have been at the other end as well, and I was part of the first departmental conference, NSPUIE, that Dr. Orkumar Mitro had organized, and he must fondly remember. This brings back memories of 15 years back when I was the first class of microbiology honors at St. Xavier's. Today, I'm going to talk about an elegant offense against first-line defense, where rheumatology meets infectious disease through an evolutionary lens. But before that, I would just give a snapshot of my academic journey, which Dr. Sudeshna just introduced. So I started off in 2005 at St. Xavier's in Calcutta, where I did my microbiology bachelor's. Then I did my master's in biochemistry in the University of Calcutta Department of Biochemistry. Then I transitioned in 2010 across seas to New York at Cornell, where I did my PhD in immunology and microbial pathogenesis. Then I did a postdoc at Yale University in the lab of Dr. Ruslan Medvedov, a pioneer in toll-like receptor biology. Finally, to chase my translational aspirations into 2018, I moved to EMD Saronno, where currently I am a senior scientist doing research in autoimmunity. Moving on to today's agenda, I would introduce the basics of COVID-19, which I'm sure all of us are familiar with currently. Then I would introduce evolutionary medicine, the basic principles of which allowed us to draw parallels of COVID-19 with autoimmunity, leading to repositioning of one of our lead drugs for COVID-19. And finally, I would recommend new age therapeutic strategies using evolution as a guide stick. COVID-19 has been a catastrophic calamity. It has claimed more than 1 million lives in the 9 months. And India and the United States are two countries that have been significantly impacted. The disease takes a toll on the elderly, especially males with comorbidities like hypertension and diabetes. This disease has totally changed our social, hygienic, and travel norms beyond imagination. Besides, the economic recession that looms has led to physical and mental anxiety-related changes in people. COVID-19 is caused by the virus SARS-CoV-2, a single-stranded positive sense RNA virus. The symptoms of this disease include dry cough, fever, anosmia, and diarrhea with difficulty in breathing. This virus is a respiratory virus and is transmitted via both aerosol and droplet, thus being extremely contagious. The preventive measures for this disease include proper hygienic practices, wearing a mask, and physical distancing. In today's world, it's very important to be even more socially connected despite maintaining physical distance. The treatment of this disease currently are under clinical investigation. Because of the unprecedented nature of COVID-19, an unprecedented strategy of global collaboration was required to tackle this disease. A fast and furious approach was taken where there was collaboration between public and private organizations, governments, academic labs, and pharma companies to come up with an unprecedented mode of testing, which is unparalleled in history. Testing via qPCR, via serologic test with antibody detection, or antigen-based detection. Preventive measures, as I mentioned, scaling up personal protective equipments, masks, hand sanitizers, and treatments with therapeutics and vaccines that are undergoing clinical investigation as of now. And the Gates Foundation, which is a philanthropic organization, has highly contributed to this COVID-19 therapeutics accelerator effort. One of the ways to fast repurpose existing drugs for a new disease allows maximal resource optimization and allows an existing drug to be used in multiple indication. This strategy called drug repurposing or drug repositioning has been used to treat COVID-19. The classic example for drug repurposing comes from those exampled by rheumatic disease, typified by rheumatoid arthritis and lupus or SLE, which is the main focus of our interest at EMD Saronno. For these diseases like lupus and rheumatoid arthritis, there have been drugs that are used as repurposed or repositioned ones. For example, an antimalarial hydroxychloroquine has been used as a treatment for lupus and RA as a mainstay for the patients for many years. Methotrexate is an anti-cancer drug that has been used also for autoimmune indications. And glucocorticoids, which are steroids, are also used broadly to suppress inflammation in autoimmune diseases. COVID-19 is a new disease, and drug repurposing is an optimal approach to tackle something of the unprecedented nature of COVID-19. However, this needs caution because this is a new virus. We hardly understand its biology much. And there should not be any rush in this desperation that compromises long-term safety of patients. Understanding the immune response to SARS-CoV-2 is of paramount importance, and rationalized therapies need to be developed, which not only address the symptoms, but actually treat the cause of the disease. Taking a step back, what is a disease? The WHO, based on its classification system, classifies diseases based on 4 broad categories. A disease, based on the topographic or anatomic classification, includes cardiovascular disease, gastrointestinal disease, liver disease, and so on, based on the organ system that is affected. Based on the nature of the disease and the pathology it manifests, diseases can be neoplastic, inflammatory, or metabolic. Based on the etiologic agent of the disease, that is the causative agent of the disease, diseases can be bacterial, fungal, or viral. And finally, based on epidemiology or statistics, a disease can be of the nature of endemic, epidemic, or pandemic like COVID-19. This classification system is very useful for clinician and physician training. However, this lacks the evolutionary reasons behind why we get sick. A nascent form of alternative medicine called evolutionary medicine integrates aspects of evolutionary biology with medicine to a field called evolutionary medicine, which is important for new age physician and clinical training, because nothing in medicine makes sense except in the light of evolution. Life history theory shaped evolution, and every aspect of our health and disease is impacted by this. When we look at diseases through an evolutionary lens, the evolutionary medicine classifies diseases into 7 categories. The first category is genetic disease that are caused by single mutations in genes. For example, thalassemia, which is caused by mutation in hemoglobin B subunit. These diseases are incompatible with reproduction but are not embryonic lethal. The second category of disease is environmental. These are major drivers of extrinsic mortality, and these are diseases primarily caused by pathogens like virus, bacteria, and fungi. Also, other diseases like those caused by toxin exposure also fall in this category. These are diseases against which we have evolved protection mechanism. For example, the immune system, the detoxification system, and so on. However, because the immune system has evolved to provide this high benefit, it is also susceptible to dysregulation, leading to diseases of defense system dysregulation, which is the third category. This is bystander cost associated with defenses like behavioral and immune responses. Examples of disease in this category are autoimmunity, allergy, and obsessive chronic behavior, OCD. The fourth category of disease is that related to homeostasis, which means these are diseases that happen due to an environmental mismatch. A simple example is that of obesity and type 2 diabetes. In today's world, due to the abundance of junk, cheap food combined with sedentary lifestyle, obesity has become an epidemic. This is related to change in the adjustable homeostatic set point, thus falls in the homeostatic category. Another category is related to maintenance mechanisms. Maintenance mechanisms are those that contribute to intrinsic mortality. Diseases in this category are like neurodegenerative diseases and cancer. These are diseases probably which we have evolved to prevent, but not to naturally cure. The final 2 category of diseases are due to stochastic random events in development or due to genomic conflicts. For today's purpose, environmental disease typified by COVID-19 and defense system dysregulation disease, autoimmunity like lupus, are prototypical examples that I will use to draw parallels. Principles of evolutionary medicine include reasons of what and how a disease is caused. These are called the proximate cause of a disease. Whereas the evolutionary cause of the disease is the ultimate cause or why we get sick. For example, fever is a response that perhaps due to infection leads to an increase in body temperature. The proximate cause of this disease is because of cytokines that act in brain centers to elevate the body temperature. But if we think from an evolutionary perspective as to why we get fever, the reason might be that the increase in body temperature perhaps protects us by increasing the resistance against the pathogen and making the pathogen survive less. Thus, before gulping down indiscriminately aspirins or paracetamol, we should perhaps think as to why we get the fever, and maybe the fever is protective. Other tenets of the evolutionary medicine principle include the fact that there are diseases that are sexually dimorphic. For example, cancer is highly prevalent in males, whereas autoimmunity is a disease which is biased towards female. An important aspect of evolutionary medicine is that of trade-off. As the name suggests, it is a feature which, when beneficial, is always constrained by another aspect which is detrimental. So a gain of function or a benefit in early life might lead to consequences or harm later in life, for example. Mismatch with the environment in time and space also impacts disease. For example, immigration to a different country with different culture, different food practices can uncover a disease in a person. For example, I know many people who, after immigration to USA from India, who had no previous history of allergies to pollens, were diagnosed with allergies to pollens in the spring season. This is an example, perhaps, of mismatch in space. Finally, co-evolution with pathogen and the arms races that we face because of coexistence and co-evolving with this pathogen is the final tenet of the evolutionary medicine principle. Pathogens evolve virulent strategies in the face of the immune defense that our body provides them. This mechanism of disease resistance is countered by the virus, which shows virulence. Pathogens can induce exaggerated defenses for their own benefit. For example, there are many respiratory viruses that purposely induce excessive coughing so that it can facilitate transmission from one host to another. Autoimmunity or allergy is a trade-off, that is a cost that we pay for the benefit, that is pathogen resistance. So loss of self-tolerance in autoimmunity, where we start to mount a response against self, is a trade-off cost that we pay because of the high benefit of disease resistance. But besides disease resistance and self-tolerance, there's another concept which is important. This is the concept of disease tolerance. This is the ability to tolerate the damage and repair and regenerate. A simple example would be comparing robustness with resilience. Robustness in the case of this boxing match is perhaps how much can you take before you fall down. This would be an example of disease resistance. Consider our bones. Our bones are very hard to break, but once broken or fractured, it's very hard to heal. Whereas resilience is how long does it take you to stand up again after falling. This is the example of disease tolerance. For example, our skin is very fragile and it gets wounded and cut very easily. However, it shows resilience and can regenerate and heal pretty fast. So this balance of disease tolerance and disease resistance defines health and disease in many contexts. In the context of COVID-19, what is becoming extremely clear is that this disease does not necessarily typify, in at least the severe cases, a lack of adequate immunity. What is becoming clear is that in severe cases of COVID-19, there is a hyperimmune response. The immune system goes into overdrive and throws the kitchen sink at the virus, and in the process causes bystander damage via pro-inflammatory cytokines and the very famous COVID-19 cytokine storm. Not just that, there are adaptive immune cells that start to make autoantibodies due to perhaps loss of self-tolerance, and they start to attack self. And such antibodies can neutralize molecules that are important for defense against the virus, like interferons, and this can cause increased susceptibility to COVID-19. What we are kind of seeing perhaps is not just loss of disease tolerance, but also loss of self-tolerance that is leading to an exaggerated response in COVID-19, which is very similar to what we see in autoimmune diseases like lupus. So this goes against the canonical view. The canonical view is that infectious diseases and immune system hyper reaction diseases are at the opposite end of the immunologic spectrum. But what COVID-19 is perhaps showing us that COVID-19 and autoimmune diseases like lupus are perhaps two sides of the same coin. And this balance of immunopathology and disease tolerance can determine the outcome of severity of the disease. SLE, or systemic lupus erythematosus, shortly lupus, is a chronic disease that primarily affects young females. In this context, pro-inflammatory cytokines like IL-6 and type 1 interferons lead to aberrant activation of T and B cells, leading to production of autoantibodies, meaning antibodies that recognize self-components, including self-RNA molecules. In COVID-19, what happens is that, again, there is a cytokine storm related with IL-6, TNF, and even interferons. And there are antibodies that are produced against the virus. And these can trigger inflammation in cells via recognition of single-stranded RNA, which is the genome of the SARS-CoV-2 virus. And this antibody-dependent immune enhancement, ADIE, can lead to clinical manifestations in both SLE and COVID that have uncanny parallels. For example, fever, fatigue, rash, and neurological symptoms are common in both diseases. And although COVID primarily affects the lung, there is evidence like affecting kidney and also brain. And these are common in a systemic disease like lupus also. Finally, most importantly, vascular inflammation and coagulopathy has been uncovered in COVID-19, and these are also seen in SLE. Another aspect which is very similar between COVID-19 and SLE is the ethnic groups that are affected. There is a demographical bias towards minority groups like African Americans and Hispanics in both SLE and COVID-19, and underlying conditions like type 2 diabetes and hypertension associate with both SLE and COVID-19. The cellular and molecular processes that underlie these clinical manifestations are perhaps similar between these two diseases, thus this uncanny similarity. For example, monocytes via inflammasome activation after sensing danger signals can release IL-1 beta. Macrophages via antibody immune complex internalization can lead to immune complex dependent activation in both lupus and COVID-19. Neutrophils can undergo an inflammatory form of cell death called netosis, which can then release neutrophil DNA, causing platelet aggregation and blood clotting defects, leading to vasculopathy. And finally, even phagocytes via complement-dependent activation can cause pathology in both these diseases. The immune complex aspect that I mentioned made us think and believe that there is an axis where in autoimmune diseases like lupus, RNA-containing immune complexes, when internalized via immune cell receptors called FC receptors or complement receptors into endosomes, lead to activation of TLR7 and TLR8, two toll-like receptors that recognize single-stranded RNA. And in COVID-19, what we think is that SARS-CoV-2, when neutralized or suboptimally neutralized by antibodies, can again be taken up into immune cells that express receptors TLR7 and 8 to sense the single-stranded RNA virus genome. This process leads to an activation of a pro-inflammatory immune cascade dependent on NF-kappa B, leading to production of TNF, IL-6, and many other pro-inflammatory cytokine, causing the cytokine storm. Thus, we hypothesize that as we have done in the context of lupus using a drug, M5049, which is a TLR7-8 dual antagonist to block this pro-inflammatory signaling, we thought that in COVID-19, if we use the same drug, we might find some benefit. And we filed an IND petition with the US FDA for the same. And what we found was there was FDA approval for our IND filing. And this TLR7-8 inhibitor, M5049, is currently in phase 2 clinical trial for COVID-19. And the name of this trial is the anemone trial because anemone is the structure, has the shape of a corona. Not just the TLR7-8 inhibitors, the entire rheumatology field has used a whole bunch of drugs from the rheumatics shelf for repurposing for COVID-19. This includes cytokine modulators like IL-6 blockade, which is pioneered by Roche. And there's also therapies which include blocking of JAK-STAT pathway with JAK inhibitors, which Eli Lilly has pioneered. We have also contributed one of our drugs, interferon beta or REBIF, for NIH clinical trial, which is the ACT-3 trial. And the reason for this, we believe, is that during the course of COVID-19, what is perhaps happening in severe disease is that in the initial phase, the innate immune response is suppressed by the virus. But later in the disease, the virus perhaps exploits this strategy, or the immune system is very naive and does not know how to tackle the virus and goes into overdrive because of TLR7-8 dependent activation, and leading to exaggerated response and finally ARDS that leads to death. So in this context, what we believe is that anti-rheumatic drugs would work best in the latter half of the disease, whereas antivirals could work best in the early half of the disease for prophylactic purposes. Moving forward, thinking about the future and rationalized therapies that are required for viral infections, we have to consider that viruses live by a balance of virulence and transmission. Transmissibility of a virus is determined by the reproductive number R0, meaning one person contracts the virus, how many more people get it from that person. And the more the reproductive number of the virus, like for example, measles has a very high R0 of 9. But in that case, the disease severity or the case fatality rate is much lower. So MERS, for example, has a very poor R0, does not get transmitted, but that might be because it has a very high mortality of 35%. In this aspect, SARS-CoV-2 seems to be optimally evolved such that it has a balance of R0 and transmission and virulence, such that it is able to perpetuate and still cause death in the context of people with comorbidities and underlying conditions. One of the limitations of immune-related therapies is that we have till now used a sledgehammer approach. For example, when we want to treat cancer, we have used chimeric antigen receptor T-cell therapy. What this has done is that although it can try to cure the cancer, but it leads to a side effect of an IL-6 associated cytokine storm. And in immune checkpoint blockade, which won the Nobel Prize in 2018, which has revolutionized cancer treatments. But the side effect of this is autoimmunity. And in many autoimmune diseases where we have used cytokine modulators like TNF alpha blockers, IL-17 blockers, or interferon 1 blockers, in that cases for these autoimmune diseases, what we see is that there's a side effect that for TNF blockers, we have susceptibility now to increased bacterial infection. For IL-17 blockers, we have susceptibility to fungal infection. And for interferon blockers, we have susceptibility to viral infection. Thus, it seems like we have to tread a fine line in rationalizing future therapeutics and not use a sledgehammer approach. An example for our new age therapeutics would be something along this example. If we try to read this paragraph here for 10 seconds, I think none of us would be able to meaningfully interpret what is written in this text here. However, if we try to read this, it might take us some time. However, if we read this, it would make some sense to us much better than the previous paragraph. And in essence, the sense that we get out of this paragraph is very similar to the actual correct text, which is written here. And the reason why I bring this up, this example is called hypoglycemia, which I mentioned here in this middle paragraph. The reason why I bring this up is because our approach to date for therapies is something like this, where we have a disease state and we want to revert it complete to the healthy state. However, what I would like to propose is because of trade-offs associated with evolutionary principles, it might be advisable to fine-tune diseases such that we come up with therapeutic strategies that are hypoglycemia-like, which work in a way to make sense and restore health, yet does not cause overt side effects. And this is the principle called the Goldilocks principle. So Goldilocks, when she was offered porridge, which was either too hot or too cold, she preferred to choose one which was warm and just right. So using the principles of feedback loops, if we design therapeutic strategies around immunomodulation, which are based on immunooscillation, meaning using combination therapies that can lead to generation of oscillatory loops such that we remain in the optimal window of health where we do not have exacerbated viral infections, nor do we have exaggerated immune responses. Such a sweet spot would be something which would be desirable for optimal health. With that, I would like to end and I would like to thank my mentors at different stages of my career. And I would especially like to thank Dr. Orkuar Mitro, who was the head of the department when I was there at St. Xavier's in 2005, and the MTIM organizing committee. And I'm happy to take any questions. Thank you.
Thank you for that very informative talk. I would now request once again the head of the Department of Microbiology, Dr. Sudeshna Sham Chaudhary, to conduct the question and answer session. Ma'am, over to you.
Thank you, Mtim. Thank you once again. So, wonderful lecture, wonderful presentation, Dr. Vaidyanathan. So, I'm happy that I got to know about the catalog of COVID-19 interventions from the rheumatic shelf, as well as the rationalized future therapeutics, the limitations of sledgehammer approach, how far the TNF blockers make susceptible to bacterial infection, and the hypoglycemia like approach. I must appreciate this, and Goldilocks principle for immunomodulation and immunooscillation. Very, very nice presentation, Bharat.
Thank you.
Dr. Bharat. Okay, so I want to just start with a question from Dr. Arun Kumar Mitro. So he asked that can a mixture of leflunomide and gamma IFN blocker used interferon blocker used in COVID treatment?
Yeah, also this is a very good question. Actually, interferon gamma blockers are in therapeutic trials, and leflunomide is actually also a drug which we use in autoimmune diseases. So the DHODH inhibitor leflunomide is approved drug, and it is being trialed in COVID actually. So it is categorized similar to dexamethasone, which is like a steroid, and similar principle based on which dexamethasone works. Leflunomide, by inhibiting DHODH, might also suppress autoimmunity, but and COVID-19 as well. But the important point here again would be the timing of the intervention, because just like dexamethasone, early intervention with steroids is not good in the context of COVID. It is better to use leflunomide or dexamethasone in the later stages of the disease.
Thank you, Dr. Bharat. Then next question is, will toxicity by heavy metals fall under environmental by Dinosauria?
Yeah, that's a good point. I think toxicity by heavy metals would definitely fall in the environmental category. And we have, this is well studied in plants, but also in humans, we have heavy metal detoxification machinery and enzymes. So this would definitely fall in the environmental category.
Thank you. The next question is, what causes COVID-19 to have a color preference more active on African Americans and on males with context to gender, by Rishi Modak?
Yeah, so this racial bias is layered. So what happens is that, as you know, COVID-19 is a disease where there is a lot of social etiquettes involved. Minority groups might not have access to personal protective equipments like masks. Hygienic practices like with hand sanitizers, washing hands might be compromised in such communities. Besides, these are the communities that are mostly affected by comorbidities like obesity and hypertension, diabetes. And thus, that could be a trigger why they are more susceptible to COVID 19.
Uh, the next question is, if any person has stem cell disease, what is the effect of COVID-19 on it, by Shubham Dubey?
Ma'am, could you please repeat the first part? Ma'am, you're on mute.
Yes. Can you hear me?
Yes.
Okay. So if any person have stem cell disease, what
Stem
is
cell.
the effect of COVID-19 on it? Stem cell disease.
Yeah, this has not been systematically investigated as far as my knowledge goes. However, the type of stem cell that has been affected is of importance. So, for example, if someone's hematopoietic stem cell is affected and the person is immunocompromised to begin with because of HSC defects, that person has a very weak immune system to begin with. And in that context, because in the early stage of the disease, a strong immune response is required, and hematopoietic stem cell defect in that context can make COVID-19 worse. However, depending on the type of stem cell under question, this could vary.
Okay. Thank you. Thank you, Dr. Vaidyanathan. And this question was given by Shubham Dev. The next question, why pandemic rate is so high for this virus, by Debashri Bortha?
Yeah, so this virus is extremely manipulative. I feel like the more we study this virus, we'll get to know more about it. But the way the virus has spread is amazing, because 80% of the infections are asymptomatic. So what happens is that young people are not affected by this disease. Children are not affected by this disease, but they are carriers. They can carry the virus and transmit it to others who are more susceptible. So what has perhaps happened is that in that initial 1 month or 2 months in China, there were a lot of asymptomatic carriers who did not know that they had the disease because they had no symptoms at all. And in that context, when they were speaking, they were contacting others like with physical proximity, they might have given this to many people. And that is how the virus from China became a global pandemic via Europe and USA. And this is again like an example of perhaps the virus's strategy, where it is balancing its virulence and its transmission capability such that it does not impact the young people, and uses the young people for its own benefit to spread to others, like elderly and the compromised people, and then kill the compromised people.
Uh, Thank you, Dr. Vaidyanathan. I think I'm just waiting for other questions to come. Um, so, can you please tell me about many more thing related to this sort of careers? So, what are
Oh yeah,
the groups who are
yeah.
acting as?
So the asymptomatic careers. So what's perhaps happening is that there are two categories in this, like so the disease falls into four different bins. So there's mild disease, there's moderate disease, there's severe disease, and critical disease. Now the carriers perhaps fall in the mild disease bin or in the asymptomatic bin. There's a special category called pre-symptomatics, meaning these people end up having mild disease later, but in the pre-symptomatic phase where they do not have overt symptoms. In that case also they can serve as carriers. And children, for example, uh, in like many thousands and thousands of children that have been analyzed, there's less than like 0.1% contraction of severe disease in children. So what is probably happening is that most children and young are saved from this disease, which is a great thing. But then what happens is we pay the cost because the elderly are compromised and they get in contact with these carriers who are younger, and they suffer a lot.
Thank you, Dr. Vaidyanathan. My last question probably. Why
Mhm.
only a certain part of infected population suffer from loss of smell and taste? So that is the marker indication for COVID that I know. So that is a loss of smell as well as taste.
Yeah, so anosmia, which is again a neurological phenotype in the context of disease, is not super prevalent in everyone. It is a certain subpopulation of people who get this. This is only being investigated now using like organoid models. So there's SARS-CoV-2 infection of the brain that has been done in vitro, and autopsies of brains of dead patients has revealed that there's a subpopulation of patients in which there is neuroinvasion by the virus. So it is not totally understood why only in some people there is neuroinvasion, whereas others are protected from neuroinvasion. So the penetrance of the anosmia phenotype is not as high as that of, say, dry cough or fever, but this needs further investigation.
This question was given by Ridhita Dev. So I'm getting a few questions. Can I ask Dr. Bharat two
Yeah,
more
sure.
questions? Okay. So, by our own student, Shwarnika Roy, she asked, how highly is it possible to get infected with the virus even if we take all the necessary precautions? Very important question.
Yeah,
How
so
highly is it possible?
Yeah, so it is almost like after a point you say it's destiny, right? But as scientists, we cannot say that. So in theory, if we take all the precautions and if we do not have underlying conditions, meaning if we do not have a history of diabetes, hypertension, cardiovascular disease, in that context, if we take all the precautions, and you don't have to be really paranoid, because there were extreme examples that were done and those are not required. If you do the basics, like hand washing, not doing handshakes, not touching your face and eyes constantly, and wearing a mask all the time, if you do those basics, it's and if you are young and in the context of COVID, it seems like females are much better off. So if you're not male, and if you're a female and you're young without underlying condition, there's a high percentage chance that you won't be affected. Even if you get the disease, it would be like asymptomatic, as is the case. Like mild disease is in 80% of the people, and asymptomatics also are around 20%. So um, uh, it's there's a great chance that severe disease would not be contracted if the basic practices are followed.
Yes, that's the thing. Another question from Dr. Arup Kumar Mitro. Can early treatment with methyl cysteine protect? Methyl cysteine.
That's a good point. I'm not very familiar with the rationale for use of methylcysteine, but if it is falling in some ways in the antiviral bin, if it has direct effect on the virus and compromises the virus in the early phase, such therapeutics do show an effect, as evidenced by success of antiviral interferon. So antiviral interferon is being used early on, and also Gilead's drug remdesivir, which has been in the limelight quite a bit, is another drug which is better off used in the early phase of the disease. But I'm not very familiar with the mode of action of how methylcysteine.
Can I interfere? Can I interfere?
Yeah,
Just
sure,
a moment.
absolutely.
Actually, the methyl cysteine the Indian Army is taking, they are saying it is mucolytic agent basically. It is not allowing the mucus to deposit, and at the same time clarifies the nasal passage.
I see.
So that particular drug they are giving so that this anosmic condition, the initial symptoms will not appear, and even if it is there, it will go off. So that's why I am asking this.
Yeah. So in terms of using a mucolytic, I think it is a valid strategy. The only thing is that, as I mentioned to you, treating just the symptoms is not the way to go for. Like if there's a rationale why the mucolysis can prevent the viral transmission, then there should be a precedence to use this. But as I said, treating symptoms, like say if you have fever and you take a paracetamol, that does not get rid of COVID in any way, and it might even make it worse. So in that context, caution should be used as to whether it is just treating the symptom of excessive mucus in the nasal passage, versus if the mucolysis with methylcysteine impacts the virus virulence per se, or the viral replication and survival. If there is basis for that, in that context, it should be used.
Another question, Sudeshna, I'm sorry.
Yes.
I want to ask this question, and because the session is ending, I just am keen to
Two
ask.
more questions are there from audience. So just
Bharat,
after
just you tell me, in a country like India,
Mhm.
honestly speaking, antiviral drugs are so costly. So you have
Mhm.
said antiviral drugs to be prescribed initially in the initial phase of COVID. So
Mhm.
can you suggest any low-cost antiviral or low-cost immune suppressor which can be actually accessed by common people?
Yeah. So as a prophylactic, even Dr. Fauci here has recommended use of vitamin C and vitamin D. So that is something, and in Indian medical practices that we have used, like, be it Ayurveda or whatever, those are aspects of prophylaxis. So if your immunity is strong to begin with, if you use agents which boost your immunity in the early phase of the response, be it with vitamins or be it with antivirals, it could be effective. Now, the point which you make about cost is a very important one, because for this pandemic to go away, we have to dissipate all the therapeutics in a way which is accessible to the poor, because most people who will get impacted by this are people who are poor, who are migrant workers, who are underprivileged, and these people will be get hit very hard. And so at least for the vaccine, there is global effort to make sure that the vaccine is cheap and affordable, and India is at the pioneer of manufacturing in large scale vaccines at a cost effective manner such that it would be given to everyone. But for antiviral medications, as you said, this has something that really needs to be addressed, and big pharma companies need to subsidize the cost of their drugs for this kind of a pandemic.
Dr. Vaidyanathan, another question once again related to the olfactory thing. That is, how long do symptoms of smell or taste persist in case of infection? How long it persists? By the
Yeah.
Datta.
So this is highly variable. So the reason for that is, as I said, anosmia, the loss of taste and smell, is not very penetrant in everyone. So because it's not penetrant in everyone, it is not exact, and because it's not exactly understood as to how and why we are getting this neurological symptom, uh, the persistence of the anosmia also varies. So what is very clear is that it's the this is a clear sign. If you have diarrhea and anosmia together, there's a pretty high chance that you have contracted the virus. However, the persistence of that does not necessarily track with like viral clearance and recovery. So what is more important in for the recovery is the partial pressure of oxygen coming back, the SpO2 saturation, and the shortness of breath. If you're recovering from the shortness of breath and your oxygen saturation is greater than 95%, then chances are you have recovered. But using anosmia as a cue for recovery is kind of challenging, just because at this point it is not very well understood the mechanistic basis for the same.
Thank you. The next question is, will COVID reinfection lead to hyperimmune responses and cytokine storm reinfection? By Anunadottu.
I think this is a very, very important question, and
Yes.
this is absolutely important for vaccines. So our hypothesis was basically that antibody dependent enhancement. So this is antibody dependent enhancement is very well characterized for Zika and dengue. So if you have dengue and then you get Zika, you have antibody dependent enhancement. So in the context of the same virus, the reinfection, if it's a slightly different strain, so for example, now there's precedence that the virus has mutated. There's a critical mutation in D614G of the spike protein S. And this mutation makes the virus sufficiently different to be called a different strain of the virus. And the second wave that happened in USA, hitting Texas and Florida, majority of the viral isolates were this new mutant strain. Surprisingly, what was found was that this mutant strain was more contagious but less infectious. So as I said, like the R0 and the CFR are reciprocal. And what seems to be happening with the reinfection with this new strain, you are not getting that severe a disease. So it seems like this hyperinflammation is probably not happening in the context of reinfection with this different strain, which could be linked to antibody responses in during the first infection. So it's possible that our antibodies that are generated and the B cells that are hopefully will have memory against the virus do take care of the reinfection. But there are cases which are isolated now with the reinfection. In a couple of cases of reinfection which happened in Hong Kong, the evidence was that the second infection was less damaging, whereas one case in the US came up where the person did get symptoms. So it is not exactly clear whether it's equally penetrant or not. It's very heterogeneous. That is why we have to keep in mind like demographics also, like what happens in Hong Kong, what strain is there versus in USA, what strain is there. Those things might impact the immune response upon reinfection also. But a very, very important question, especially for vaccines.
So just a moment. In that case, what you are saying, that means we will need booster dose of vaccine.
Absolutely.
Because antibodies will go
Absolutely.
away in
So
2-3 months.
100%. So this coronavirus, the immunity lasts not more than 3 to 6 months. So even if we get a vaccine, and hopefully we'll get one by the end of this year, at least the phase 3 clinical trial of Pfizer and Moderna would be out. But for sure, we would need a booster dose. But what would be different from flu is that in the case of flu, we also get every year flu shots. But for flu, it's a different strain and a different vaccine prep every year. But hopefully for COVID-19, it would be just one vaccine, which we would need multiple doses annually, maybe.
Thank you. And this is the last question. Why is there a sudden increase in recovery rate in these cases? By Stuti Borua. By Stuti Borua, why is there a sudden increase in recovery rate in these cases?
So one interesting point about recovery rate, because I have been tracking the statistics of COVID-19, particularly in India and USA very closely. What I found was that in India, there is a very high recovery rate. And again, the exact reason is not known, but the differences in recovery rate between different countries can again go back to this point that the disease disparately affects people who are male versus female, which I mentioned again, sexually dimorphic, and young versus old. So in India, the population, we have a very young population overall. So compared to other countries like Japan and Italy, where there are a lot of people above 60 and above 80, but in India, population at large in 1.4 billion is younger, less than 50. And so because more than 80% of the diseases is mild and in the young people, so there is a chance that because of that, the recovery rate in countries like India with a younger population is more than other countries which have a significantly higher elderly population.
Thank you, Dr. Vaidyanathan, for these wonderful interactive sessions.
Absolutely. These questions were extremely motivating. I think like the students, the professors, whoever asked these questions are very, very insightful questions. And it was great to interact with all of you. Thank you again for this invitation and all the organizing committee members, because this takes a lot of time. I've been there. So great job, everyone. And thank you, Arup sir and Sudeshna ma'am, for this.
Thank you for your wonderful presentation.
Ma'am, I would now request you to present our speaker with e-bouquet and an e-memento and an e-certificate.
So Bharat, this is for you. Thank you very much for such a nice presentation and nice interaction. Thank you, Bharat. This is for you.
Thank you. Thank you very much. It's very,
It's our
very
honor
humbling. Very humbling.
to have you as. Thank you very much.
So that brings us to the end of our first technical session. I hope to see everyone in the second technical session from 1 o'clock. And for our dear spectators, please keep an eye for the feedback link because it might be shared at any point among the 3 technical sessions that will be happening today. And until then, please take care and bye-bye.
All right. Thank you, everyone. Bye.